Right, as we continue a little bit our review here, we do need to talk about the cell membrane and also go into some detail about the transport mechanisms across the cell membrane. Some of these will be review, but some of these will be more detail than you have studied before with regards to these mechanisms. So the cell membrane, as you recall from biology, is what regulates what enters and leaves the cell. So it is responsible for maintaining the homeostasis level of the cell itself. We describe the cell membrane as being semi-permeable. In other words, some substances are able to enter or leave, but others can't. In general, though, we put nutrients in and take waste out through the cell membrane. It provides protection and support for the cell. So we're going to focus on the substances entering and leaving the cell, and this is what we refer to as transport. There are two main types of transport across the cell membrane. The first type is passive transport. And if you think of passive, it means it's not really do anything. If somebody is passive, they're really not expending any energy, and that's exactly what this type of transport does. The type of passive transport that you are most familiar with is diffusion, and you actually studied this in biology. Diffusion goes from areas of high concentration of substances to low concentration areas of substances. And just like we said, because it's passive, it requires no energy. Now, this diffusion will happen until the molecules reach what's called equilibrium. In other words, equal concentrations on both sides of the membrane. However, it's not like the movement actually stops at that point. What happens is they continue to move across the membrane, but it's in equal numbers to maintain the concentrations. Now, diffusion is dependent on a couple of characteristics of the substances. The first one is the lipid solubility of that substance. In other words, can it dissolve in a fat? The cell membrane itself is fat. It's made up of a bilipid membrane. So if it's able to dissolve in a fat, then the more likely it's going to be able to diffuse. The second factor is the size of the substance. Substances that maybe aren't lipid soluble but are able to but are small are able to diffuse very easily across the cell membrane. Other substances which maybe are lipid soluble, but because they are so large, they cannot freely diffuse. They have to use a different transport mechanism. Now, one of those small molecules is water, and water actually is able to freely diffuse across the cell membrane. But we don't just call this diffusion of water, we call it osmosis. So, osmosis is that diffusion of water. Like I said, the cell membrane freely allows osmosis to occur. So it can't keep water out, and it can't prevent water from being lost from within the cell. That means that cell membrane and the environment around the cell is very, very important for maintaining the homeostasis of the cell, since many of our substances within the cell have to be dissolved in water. So we can have our cell be placed in different solutions, and depending on how those solution concentrations are, it will affect the osmosis of the cell. If we describe something as being isotonic, it means that it is the same strength of the solution inside of the cell as outside of the cell. We would see no change in the cell taking place. In other words, it would just look like the cell is actually sitting there. Diffusion is occurring, but it's occurring equally in both directions. If I describe a solution as being hypotonic, it means that there is less water on the inside of the cell than on the outside of the cell. And what we would see is, if we are actually looking at the cell would be swelling because the water is diffusing in. The last situation is hypertonic, where I have more water on the inside than I do on the outside. And what would happen is I would actually see the cell shrink or shrivel as the water diffuses out. Now, this may not seem like a big deal, but it can have a huge impact on the functioning of certain cells. For instance, like red blood cells. Red blood cells have to have this kind of concave donut, jelly donut, or snow tube type of shape with this little divot in the middle. If the solution that they are in, let's say I'm given an IV solution of the right concentration, that's the same concentration as my fluid in my blood, then that's isotonic, and my red blood cells won't change their shape. However, if instead I'm given a IV solution with, let's say, pure water, distilled water, then that is a hypotonic situation. And you can see here by the arrows, the water would actually diffuse into the red blood cell and really change its shape. This shape change actually prevents that red blood cell from carrying the oxygen like it's supposed to. So this red blood cell will not function correctly. The last situation would be if I got too strong of a solution, so it's too concentrated. The water would actually diffuse out of the cell, and you can see my cell would shrivel. Again, it would not be able to carry oxygen in that shape. In addition, as you can see, we've got kind of some bumps and grooves there. It would be more likely to get caught along the blood vessels and actually form clots within my body, and that's something we want to try to avoid. Now, the rest of the types of transport that we have fall into the category of carrier-mediated transport. In other words, diffusion and osmosis happen just on their own, right through the membrane. But sometimes we can actually move things using the help of the membrane proteins that lie within the cell membrane. So what happens is these membrane proteins bind to specific substances and carry them across the membrane. These substances fit together like an enzyme and a substrate, so they fit together in that lock and key type of mechanism. The shapes have to match. And this carrier-mediated transport can be active or it can be passive. So we want to talk about the passive type of carrier-mediated transport first. The passive type of carrier-mediated transport is called facilitated diffusion. And what happens within facilitated diffusion is we have protein channels that help move molecules across the membrane. The molecule binds to a receptor site, and then the protein shape changes. The molecule is then either released inside of the cell or outside of the cell, but we have no energy that is needed. A typical substance, and a very common substance that has this happen, is glucose. You can see here these purple hexagons are our glucose molecules. They come in and bind to the receptor site on that carrier protein. And when that happens, the carrier protein changes shape, and then the glucose is released to the inside. The membrane protein will then change its shape back so that it can repeat that process with another glucose molecule. This type of diffusion is often confused for active transport. What you have to keep in mind is no energy is required with this. Now, all of the rest of the types of transport that we're going to discuss are called active transport, or they fall within the category of active transport. Active transport goes against the concentration gradient. In other words, it goes from areas of low concentration to areas of high concentration. It's kind of like the opposite of the passive ones. These type, because they are active processes, require energy in the form of ATP. We have some examples, and we're going to go through most of these today. One of these we will actually see specifically within one of our body systems. Endocytosis, Phagocytosis, Exocytosis, and the sodium-potassium pump. Now, the types of active transport that we are going to deal with fall in the subcategory of vesicular transport. Basically, this means that we are moving materials out of the cell or into the cell in a structure called a vesicle. These are small sacs that form or fuse at the membrane. And we have two major types that we're going to go through: endocytosis and exocytosis. And if you think about what those names might mean, those prefixes of those names, endo means to bring in, and exo means to come out. So, we're going to deal first with the endocytosis. And like I just said, with endocytosis, the materials outside the cell are packaged to import into the cell. We have 3 types of endocytosis that we're going to go through, and what you need to keep in mind is that all are active processes and require ATP. So the first type of endocytosis is receptor-mediated endocytosis. With receptor-mediated endocytosis, we have target molecules that we want to bring into the cell that are present in high concentrations. Along the membrane surface, we have receptors, and these receptors are specific to those molecules. Again, they fit together like that lock and key, and they bind together. As those receptors are binding those target molecules, the membrane starts to form a pocket that gets deeper and deeper and pinches off eventually inside of the cell to form the vesicle. Examples of items that our cells bring in using receptor-mediated endocytosis include iron and cholesterol. So if you look at the diagram here, you can actually see this taking place. These little almost look like plankton shaped things here at the surface are the receptors. And what this diagram is calling the ligands, or what I'm calling those target molecules, whatever we're looking to bring into the cell. They bind with the receptor here, and you can see the shapes match, and we get this formation of this pocket which deepens. And you can see here, by just bringing and pinching off here, we get a vesicle. The cell then goes and pulls off those target molecules that it wants in the cell off, and then it pushes the vesicle back up to the surface with the receptors intact so the process can repeat. Now this is the most complicated form of the receptor of endocytosis. So if you take a look at the next type, pinocytosis. Pinocytosis means cell drinking. And the reason it's less complicated is no receptors are involved. Basically, the process is the same if you look at the diagram from before, we just don't have the receptors present there. The vesicles form at the surface and simply take in that extracellular fluid and whatever is dissolved in it. So this process is a lot less selective than the receptor-mediated type. The last type of endocytosis is a process called phagocytosis. And phagocytosis is called cell eating. And the way I learned it, that's kind of stuck with me over the years, is I call it the Pac-Man effect, because it's kind of like Pac-Man and going and gobbling up all those little dots in the game. It works very much the same way. Basically, when we produce vesicles here, it's going to contain some sort of solid object, and the solid object can be quite large, even as big as the cell itself. We see phagocytosis a lot as part of our immune response to foreign objects. And that is actually what is shown in this diagram here. We've got a foreign object here, and you can see as the cell comes near it, it sends out these extensions of the cytoplasm. And you can see it kind of looks like it's coming around like a mouth to chomp it here in number 2. That's how the vesicle forms. Then whatever is inside actually gets destroyed, and then we release that undissolved residue back out into the extracellular fluid, which will eventually get filtered out. So those are the 3 types of endocytosis. Exocytosis, actually, we've kind of seen in some of these diagrams, but we just haven't really talked about it a lot. It is the reverse of endocytosis. So what happens is the vesicle that is in the cell fuses with the cell membrane and discharges whatever contents it has outside of the cell. For instance, with phagocytosis, when that undissolved residue is released, that is actually the process of exocytosis. Exocytosis is very important to cells as they eject things like hormones, even something as simple as mucus, as well as waste products. In the next section, what we're going to do is we're actually going to get started with our discussion on tissues.